In Chapter 6, we're going to take a look at viruses, kind of look at what are they made up of, how do they reproduce, how do they get into various cells. That's everything about viruses, but then we're also going to briefly talk about prions, and I'll talk about why we talk about prions in the same chapter we talk about viruses. So we're going to start off in this chapter taking a look at what are viruses composed of. So what are the parts that make up a virus? We're going to talk about how they can attach to different cells using different proteins, how after they get inside of a cell and take it over and reproduce inside of it, how they then exit the cell to go find new cells to infect, how we can grow viruses, because they don't grow the same way that bacteria do, and then we're going to talk about prions in this chapter. And again, I'll talk about why we talk about them together. So just some basics of viruses right away. So we don't know exactly when viruses came about. We know that pretty much after life started, viruses started along with them. And so they have played a role in the evolution of anything in the bacteria, the Archaea, and the Eukarya domain. We also know that they are intracellular parasites. They have to get inside of our cells, and they're going to cause damage. They're going to cause some type of destruction. Now, in your book, in table 6.1, this is all the properties of viruses. There's a lot of properties of viruses. I'm going to try to shorten it up a little bit. One, yes, they are intracellular parasites, and they can affect anything alive. They are extremely small. So they're in the nanometer size, so our regular light microscopes would never be able to see them. They're not a cell, so they don't reproduce like cells. They aren't a cell. They don't have all the organelles like cells. And partly because of that, they don't fulfill the main characteristics of life. They can't reproduce on their own. They can't react outside stimuli. They aren't a cell. They're only active once they get inside of our host cells. They're made up of proteins and nucleic acids. The nucleic acid that they have could be DNA or it could be RNA, but they don't have both. And that DNA has a lot of variety. It can be double-stranded DNA, single-stranded DNA, single-stranded RNA, or even double-stranded RNA. A lot of variety. The molecules that are on their outside surface gives it that specificity for knowing which host cells that they can enter and attack. Once they get in, they hijack our host cells, they hijack our own DNA, and our cells, or any cells that they infect, start to become virus-producing factories. They lack enzymes for most metabolic processes. Again, they use all of our cells, and they lack machinery for making their own proteins. They, again, hijack our cells. Now, when we talk about their size, they are small. They're generally less than 2 micrometers. They're, again, usually in the nanometer size. So we need an electron microscope to be able to see these. So just to give you an idea of size, so here's an E. coli bacteria, here's a streptococcus bacteria, another bacteria, then we start to get in the viruses. And there are a couple larger. There's one called a megavirus. The pox virus is one of the biggest one that affects humans, like size-wise biggest ones that affects humans. But otherwise, we get all the way down into these tiny, tiny viruses. They're extremely tiny. If this is a bacteria and this is a virus, we were so happy to see bacteria after we had microscopes. We weren't going to see something like this. Another picture just to show comparison. Here's a red blood cell and an E. coli right next to it, just to compare a eukaryotic cell to an E. coli bacteria. And then you can see next to the bacteria, we zoomed in on the bacteria, the size of some of the viruses that can cause human disease or some of the ones that are out there that can affect plants too. But we're going to zoom in of these. And again, smallpox, one of the largest human viruses, but then we get into some of the tiniest ones like polioviruses or some of the viruses that infect bacteria. They're so tiny. If here's an E. coli, which again, we get a magnified a whole bunch just to see underneath the light microscope, we're not going to see this thing, and definitely not in any kind of detail. So, what makes up a virus? Well, it's going to have some type of covering. All viruses are going to have an outside coat called a capsid, and I'll talk a little bit more about capsids coming up. Some have envelopes, some. If a virus does have an envelope, we call it an enveloped virus. If it doesn't, we call it a naked virus. But they all have a capsid, and they're all going to have a central core where we find the nucleic acids. So that's where we're going to find the DNA or the RNA. So this is just showing an example. Here's that outside capsid, and the inside we have the nucleic acid, DNA or RNA. That's all a virus is. It's an outside coat and some DNA or RNA, some genetic material. Again, some have an envelope, but they don't have to. They really just have proteins that are found in the capsid, and they have some type of genetic material. So all viruses do have capsids, and because a virus is made up of a capsid and a nucleic acid, here's your outside capsid, and here's your nucleic acid, we call this structure, we smush the two words together, and we call it a nucleocapsid for your capsid and nucleic acid. Now again, some viruses do have an outside envelope as well. So here's your genetic material, nucleic acid, the capsid, but some also have an outside membrane or an outside envelope. Again, we call them enveloped. If they don't have it, we call them naked. Now, we'll talk about how they get the envelopes if they have them. But the capsid part of a virus, that outside protective shell, is made up of identical little subunits called capsids. And so it's made up of protein. So each one of these little tiny hexagon tan structures, that would be those protein capsids. We put them all together and it makes that capsid. Now, the capsid itself can have lots of different structures, but there are two main shapes, and then there's some variation among them. One main shape that a capsid has for viruses is it has this helical shape. It again just has that twisted turn like the old telephone cord, but it makes it look like a nice long rod. That would be the the capsid shape, and the nucleic acids inside of there. Another shape that the capsid could be is an icosahedral shape. So it's a 3D dimensional polygon. Now again, there's some variety in how many sides it has, but they can have this kind of icosahedral shape, or they can have a helical shape. And again, there's some variety in those. Now this is just to show some of the icosahedral shapes. Again, they can have some cool shapes to them. Some of them will still have an envelope on the outside, but it does have unique structures to it. Now again, some viruses have an envelope, but not all of them have an envelope. Now, if they do have an envelope, and this is mostly viruses that infect animals, that includes us too, versus bacteria or plants, that it acquires that envelope when it leaves the host cell. It steals a little bit of our phospholipid bilayer. And so here's a zoomed in picture of the envelope. This is a phospholipid bilayer with various types of proteins on the outside, these little spikes that stick out. We can see the little spikes that stick out. But that's a phospholipid bilayer. Our cells are phospholipid bilayers on the outside. So when it leaves our cells, it steals a little bit of the phospholipid bilayer, and it makes some of its own proteins. I'm like, it just steals it. Now the functions of having a capsid and having an envelope, the biggest one is protection. Again, that nucleic acid on the inside, that is the important information, just like our DNA is the important information. So it has to have that outer protein capsid coat to help protect that information. Now, it can also use that capsid, or if it has an envelope, it can use the envelope to help it find host cells. Part of it are some of the proteins found on the capsid or proteins found in the envelope. It can recognize very specific host cells, because not every virus can affect every cell that it comes along. It has to find very specific cells using its protein structures. Now, some general structures, some complex viruses. There are a couple atypical viruses. Well, the pox viruses, they are larger. They're one of the largest viruses that can infect humans, but they don't have a normal typical capsid. Instead, they have a lot of lipoproteins on their outside. And another unique type of virus are the bacteriophages. Now, the name can be confusing. You hear the word bacteriophage and you think it's a bacteria because it's got bacteria in its name. Instead, it's not. A bacteriophage is not a bacteria, it's a virus that infects bacteria. So a bacteriophage is a virus that infects a bacteria. So it's a virus. A lot of times it's confusing, so we a lot of times just leave off bacteria out of its name. So if you ever hear phage viruses being talked about, know that a phage virus is a virus that's going to infect bacteria. Now, the bacteriophages, I think they look like the coolest viruses out there. They look like something that should be landing on the moon. They still have a capsid, they still have their genetic information, but they usually have this cool tail and little tail fibers. They use those tail and tail fibers to attach to host cells and use them to inject the DNA or the RNA into the host cells. But I think they look like they should be something landing on the moon. Now, just looking at this particular structure, would you describe this particular virus, or how would you describe it? So again, we're looking at the shape of the capsid. Is it helical, icosahedral, or some complex shape? And just based on the picture, does it have an envelope or not? So is it enveloped or naked? Well, this one, it is a helical shape. Sorry, the picture didn't go. It is a helical shape and it's naked. It doesn't have an outside envelope. It's just that helical shape to it, and it's going to have that nucleic acid on the inside. Now, the nucleic acid that it can have. Again, it could be DNA, it could be RNA, they could be double-stranded, they could be single-stranded. There's a lot of variety in nucleic acid when it comes to viruses. But it's still nucleic acid and has all the information, all the genes to know how to hijack host cells. Now, the number of genes can vary depending on the virus. There can be a few genes or there could be hundreds of genes. Just depends on the virus and how complex it is, what's needed for hijacking host cells. Now, DNA viruses, usually double-stranded DNA. Just like all of our DNA is double-stranded, most viruses that have DNA is also double-stranded, but some of them can be single-stranded. Some of the DNA can be circular, and some of it can be linear. So there's variety there. If a virus has RNA as its genetic material, it's usually single-stranded, like RNA is usually single-stranded in us. It's always single-stranded in us. And so most RNA viruses have single-stranded RNA, but some can be double-stranded as well. There's, again, a lot of variety. Now, even having single-stranded RNA, which is common, there can be variety in that. There are two sides to RNA. Just like there's double-stranded DNA, if there's an A on one DNA strand, it would pair up with the T on the other. Well, RNA, if you only have one copy of information, there is technically an opposite strand as well. And so we call these single-stranded RNA molecules either positive-sense or negative-sense. A positive-sense strand, so all the A's, the U's, the C's, and the G's, and I'm like, if it has the positive-sense strand of RNA, it is ready for translation. Ribosomes can recognize it. It's just like messenger RNA. Ribosomes can recognize it, they can read it, they can translate it out and make proteins. If, however, a virus doesn't have the positive strand, it has the opposite information. That just means they have to make a template. They have to take all their negative strand information, the A's, U's, C's, and G's, and then have to make the opposite strand. So if the positive had an A, the negative would have a U. If the positive had a C, the negative would have a G. They just have to make a template first, and then they can go through translation and make all of their proteins. So how we classify the viruses. So there's lots of different things that they can have. There's a lot of variety. They can have variety in their shapes. They can have variety in their genetic material. They can have variety if they have envelopes or not. There's a lot of variety, and so we use that to group them. And so we group them into families, we group them into genre. We still italicize them. Anything that is grouped as a family, we ended in the suffix viridae. Anything that we have identified down to a genre, still italicized, and it ends in the word virus. Now, just some things to help us group into different families. The type of capsid, that's the shape. Does it have single-stranded or double-stranded nucleic acid? Does it have an envelope or not? We group some of these things by size. We group some based on what host cells they get into. Some we go based on microscopic appearances. The rhabdoviruses, this is the viruses that cause rabies, they actually have a bullet shape to them. So they have a very unique outer shape. We group them based on anatomical or geographic areas we find them. We can group some of them based on hosts. So there's lots of ways we group viruses. Kind of what it's made up of is some of the first ways we do group them, though. Now, we talked about what it's made up of, and we briefly mentioned that it does have to hijack a host cell. Well, it does, because then that hijacked host cell is what reproduces the virus. And it is a 6-phase or 6-step process. Now, animal viruses, so these affect all animals, including us, do reproduce and have some slight differences than viruses that infect bacteria, the bacteriophages. It's very similar phases, but there's a couple modifications. So we're going to go first through the basic steps of animal virus multiplication and reproduction inside of cells. So there's adsorption. This is the binding of that virus to a host cell. So adsorption is that binding. It's almost like adhesion. Then it has to get inside. So that genetic material, the genome, gets inside the host cell. The uncoating, so if it had the capsid, if it had the envelope that was all brought inside the host cell, you got to get rid of all that. Don't need it. The synthesis, you're going to make all the components of the virus. Assembly, you're going to put them all together. And then release, all those assembled viruses are going to get released out to go find new host cells. So that's the 6 basic steps. So we're going to go through, I've got a couple pictures of that in your book, because I know it's kind of small writing. In your book, it's figure 611. But you have the absorption, you have that attachment. It uses those little protein spikes to recognize specific cells. It attaches to them. It gets inside. Now sometimes the whole cell, envelope, capsid, everything gets inside of our cell. And so it sticks, it gets inside, but because all it needs inside of the cell is that nucleic acid, it gets rid of everything else. That's the uncoating. It gets rid of the envelope if there was one. It gets rid of the capsid that was there. Then we have the synthesis. It's got to make the parts of the virus. Now the parts of a virus is proteins. That protein coat the capsid and the nucleic acid. So it's got to make any spikes. It's got to make all the capsimers. That's going to make up the capsid, and it's going to make any new genetic material. So you're going to make all the parts, then we're going to assemble them so it actually looks like the virus, and then you're going to have the release. It's going to leave the cell and go infect other parts of the cell. So just looking at the different parts individually, because that's a nice overall what's happening, that you have the adsorption, that's the sticking. And it is specific for different cells. It uses those protein spikes on the outside to recognize specific host cells. Now, what cells it can affect is considered your host range. Example, hepatitis B only affects human liver cells. Hepatitis B is not going out and affecting every cell in your body. Polioviruses only affect primate, intestine, and nerve cells. Rabies affects various cells of lots of different mammals, and so it has a larger host range. And so some are very specific, some are a little more broad. Once it attaches, it's got that penetration and uncoating. So it can get inside. Sometimes it does this through fusion. Is the cell itself, when it sticks, all of the phospholipid envelope that it has, along with our cell membrane, all fuses together. It's all phospholipids. They kind of just meld together, and the protein and that protein coat the capsid and the nucleic acid come in. Then you have the uncoating. Some you have endocytosis. It's kind of like phagocytosis, that our host cell, after it attaches, goes out and completely engulfs that virus. Envelope, capsid, nucleic acid, and all. Brings the whole thing in, then you're going to have the uncoating. We got to get rid of it. A few cells can do what's known as direct penetration. That just means that once after they attach, Nothing goes inside of the cell except the genetic material. So a few can do that, but most, either all of that envelope fuses together or you have the endocytosis. Now, once you've got that genetic material inside of the cell, you have the synthesis. You have to make the parts. Well, you have to make DNA, and you're going to have to make RNA. If it's a DNA-containing virus, all the new DNA is made in the nucleus. If it's an RNA-containing virus, all of the RNA is going to get made in the cytoplasm. It's where you find DNA, and it's where you find RNA in our cells. If it's an RNA virus, the positive-sense strand that's made is ready for translation. If it makes the negative sense, it has to get converted into the positive-sense strand. It has to make a template, because from that template, we then make the proteins. We have that translation happening. The ribosome is going to read it, translate it, and make proteins. Those are the proteins that are going to become the capsid. So we're going to make the parts. So we're going to make the DNA, and we're going to make the proteins that make up the protein coat. After we've made all the parts, so just looking at this here, we've got all the nucleic acid and we have all the parts. You have assembly. We have to put them all together. We have to make it actually become a virus. Once we make the viruses, then they get released. Now, there are different ways that they can get released. One common way for animal cell viruses, especially if they're going to be enveloped, is they get released by budding or exocytosis. And this is where it steals the envelope from our host cells. So here's your completed virus, the capsid, the nucleic acid in there, but it needs to get some of the envelope. Now, there are already some viral proteins that were made and put on the outside of our cells. And when that virus leaves, it forms a bud and it steals our phospholipid bilayer as well as its own proteins, those spikes, and it leaves. And so I'm like, it just forms this little tiny bud and it leaves. Now, what's nice is the cell is not immediately destroyed when this happens, which means our cells truly become an amazing virus-producing factory. Viruses don't want to kill our cells. Then who's going to make more viruses? And so by budding and exocytosis, we make really good virus-producing factories without causing damage to our cells, at least right away. Eventually, it can cause enough damage to our cells that they die. Another way they get released is cell lysis. Lysis means break apart or rupturing, that truly these cells just burst open from our cells. Our cells die, but in the process, before they died, they made a whole bunch of viruses. And so some cells leave by budding, some cells leave by the cell bursting open. Now, concept check, little thing. Review, viruses commonly contain both DNA and RNA. That's false. They're going to contain one or the other. They don't contain both. Now, when our cells do become virus-producing factories, there's going to generally be some kind of damage to our host cells. It's called the cytopathic effect. It's damage to our cells. Sometimes it's disorientation, of individual cells. Sometimes it's a change in shape of our cells, a change in size of our cells. Sometimes they can all fuse together. So normal cells are this size. Instead, they all form together as like one big multinucleated cell. It's called a syncytium. Some will have all these additional inclusion bodies in our cells that came from the viruses. So our cells can change once they're infected. Now, this is looking at some of the changes that can happen inside of our cells. This is lots of different viruses that cause differences to cells, but some the cells lyse, some they clump, some they form those large multinucleated syncytia, some become a little more round. So there's differences or changes that can happen inside of our cells. Now, some viruses, their job is, yes, to reproduce inside of our cells, leave our cells, go find more cells, but there are times that some of these viruses can remain dormant. They're not actively going out and finding new cells to infect. They're not actively destroying cells. Now, when they're just kind of hanging out or laying low, it means we have a persistent infection. The cell is going to harbor the virus. So the virus is inside the cell, but the cell's not going to die right away. It could be slowly producing viruses, or the virus itself can actually just remain dormant inside of our cells. Now, this dormancy can last for weeks or even a host's lifetime. And a lot of times these viruses can become reactivated for lots of different reasons. A couple examples where these cells can remain active in the body. The measles virus can hang out in the brain for lots of years before all of a sudden becoming reactivating and causing issues. The herpes simplex virus is that causes cold sores and genital sores. Once it's in the body, it's there forever. It's in the body for your entire lifetime. It goes dormant and then it becomes reactivated. It's why all of a sudden you get a cold sore one day and then it goes away. Well, the virus didn't go away, but it went back into a dormant state. The herpes zoster viruses, which causes chickenpox and then shingles. If you had chickenpox, that virus is in your body, it's dormant or latent, but it can become reactivated again later on in life and cause the condition known as shingles. So it's a persistent infection because it doesn't go away. Instead, it just kind of becomes dormant, or what we call as latent. Now, viruses that get inside of our cells, other than causing damage to the cells, can actually cause cancer. You have viral DNA getting inside of our cells, and some of that DNA incorporates up into our DNA. And we've already talked about things that can cause cancer. It's anything that causes cancer is any change to DNA. Well, if you have new DNA getting added into our DNA, that's a big change. And so there are viruses that permanently alter our DNA. Well, if they're going to alter or change our DNA, you just did a mutation, and these viruses are called oncogenic or cancer causing. And it's because they're going to have an increased rate of growth. There's more of a chance for changing chromosomes. They have the capacity to divide and nothing to tell it to stop, resulting in tumors. Now, a couple common viruses that it can initiate tumors and cause cancers. The papillomavirus is a virus that causes a cervical cancer as well as other cancers. The Epstein-Barr virus can cause Burkitt's lymphoma. These are viruses that are known to cause different cancers. Now, we can call this what's known as the oncogene theory of the induction of cancer. It's long and worded of how can viruses cause cancer. So if this is your normal DNA strand at the top, our cells have built-in genes to stop cancer from happening. So we have genes to stop cells from dividing. So we have a gene, it's called a proto-oncogene. It does have onc in it. Anything oncology, whatever, anything has to do with cancer. Well, this is a gene that promotes cell division. That's what cancer is. It's cells dividing. However, the pro in front of it means it's in an inactive state. And so we have a gene that says, sure, let's divide, but it's in an inactive case. And so it's repressed. So where cells are not going to divide. We also have genes that just repress or stop cells from dividing. Again, we have all these things that repress or stop cells from dividing. So normal DNA, no cancer. However, if you get a viral infection and that virus DNA inserts somewhere near that proto-oncogene, it can activate it. And so now we have a gene, we have DNA that's saying, let's divide. That's cancer. However, as long as we still have other genes that say to stop or repress cancer, to stop or repress that cell division, we still don't have cancer. We have checkpoints. However, if another virus or if that same virus inserts into two different places in our DNA, we can activate that oncogene to divide. And if we somehow insert or change that repressor gene, it can't repress anymore. So there's now nothing to cause our cells to stop dividing. That's what can cause cancer. That viral DNA inserts into our DNA, and if it inserts anywhere near those genes that control cell division, the virus can cause cancer. Now, enough with animal viruses. We're going to take a look at bacteriophages. These are viruses that infect bacteria. Now, the most widely studied ones are the viruses that infect E. coli. They have more of the complex structure, the ones that look like they should be landing on the moon, and they do have DNA as their genetic material. Now, the multiplication goes through similar stages. It's going to go through the absorption. We're going to go through them like the penetration, the replication, the assembly, they mature. And I'm like, but the assembly and the release. It's got very similar steps. The only part is only the nucleic acid enters into the cell. The whole thing is not going to go in. Just the DNA is. And the release, it's almost always through the lytic cycle, that lysis, the bursting open of the cell. So it's very rare that a bacteria just buds and sends out new viruses. That infected bacteria is going to die. So again, the steps, it's going to have absorption. It's the binding or the sticking of the virus to a bacteria. We have penetration, that genetic material is going to get in. We're going to have replication. You've got all the parts getting made. You're going to assemble it so it looks like a virus. Then it usually has to have kind of almost like a pause where it does its full maturation into an active virus. And then lysis and release. It's going to break apart, burst open, and release all of those new viruses. They're the virions. Occasionally, though, the virus can enter a state of what's known as lysogeny. It's when the viral DNA does become part of the host DNA, and it can remain almost in a dormant state, even in a bacteria for a while. So, again, it has some of the same basic steps. We're still going to have attachment. We're going to have entry again. The only thing that goes in is just the genetic material. It's going to have all of the synthesis. We're making all the parts. We're going to have the assembly of all of the parts. It's going to mature for a little bit, and then you're going to have that release. And so you're going to have that bursting open of all of the cells. Now, again, this is just to show it's going to have the same basic steps for a virus that infects bacteria versus a virus that infects animals. Same basic steps. Some slight changes in how they get released, or some slight changes on that penetration on what actually gets inside of the cell. But if a cell can go into lysogeny, it doesn't kill the cell right away. It doesn't just start massive, I'm going to go back to my thing. I'm like, do assembly of all of these viruses, and it doesn't go through that lytic. Instead, the virus is going to become, the genetic material is going to incorporate and become part of the bacteria's DNA. Now, we call this infected cell, where the virus DNA has become part of the bacteria DNA, a prophage. And so that prophage, that viral DNA is retained, and as the bacteria reproduces and makes more bacteria, it reproduces the viral DNA as well along with it. Now, at any point, it can go back in and start the whole, continue the process. And I'm like, I like my picture better. So here's the normal lytic cycle. Ignore all this right now. Normal lytic cycle, you have attachment, you have that entry of the genetic material, you have the synthesis of all the parts, you have the assembly, and then you have the release. However, there are some that go into this lysogeny stage, where after you have the entry of the nucleic acid, that viral DNA becomes part of the bacterial DNA. And as the bacteria do what they do best, they reproduce, each of the new bacteria has part of that viral DNA in it, that little prophage, that viral snippet of DNA. And so instead of having one bacteria with viral DNA, we could have 4 or 8 or 16 every time it keeps dividing again and again. Now, at some point, something can trigger these bacteria that all have part of viral DNA in them to go back to the lytic cycle. It's a process called induction. So they go back to the lytic cycle. That viral DNA is now used to make all the parts, assemble all the parts, and then go back into release. So eventually that induction results, it activates it, and then it goes back to making new parts. Now, what can happen though sometimes is that this bacteria, which has part of a virus inside of it. It has part of viral DNA inside of it. It can actually change the bacteria a little bit. The bacteria's got new DNA. Well, depending on what's in that DNA, it could act a little different. And there are some bacteria that once they're infected with a virus, depending on the virus and depending on the bacteria, these bacteria can make toxins that they never used to make. You might have a bacteria that grows a flagella that never used to before. And it's called lysogenic conversion. And so all of a sudden you have bacteria that are making toxins, enzymes, or something else, and it's just because you have that bacteria DNA. You have those different genes found in the bacteria. Now, I kind of like this as a comparison, and it repeats, and I'm like, the lytic cycle. Let's start over. The lytic cycle, there's your virus. It injects the DNA inside of the bacteria. You have that assembly, maturation, and release. Lytic, that's the lysis. However, in the lysogenic cycle, when a bacteria uptakes new viral DNA, that viral DNA becomes part of the bacteria DNA. And as the bacteria reproduce, so does the viral DNA. And then they wait, as it will say. They wait until they go back into the induction stage, and they will continue and finish out the lytic cycle. Now, little concept check. Which of the following is a step found in animal virus multiplication, but not in bacteriophage replication? And that's the uncoating. And that's because in a bacteriophage, they only inject their DNA in. They don't have that full engulfment of the whole thing, capsid, envelope, or anything else. They just inject, so they don't need to uncoat. There's nothing to uncoat. Now, when we try to grow and identify animal viruses, again, this is going to happen. There's lots of viruses out there, and we need to identify them in patients. Well, we can isolate and identify viruses in clinical specimens. We could take a sputum sample, blood sample, look specifically for the viruses. It can take a long time, and long enough that we don't want to wait. And so, and I'm like, we usually are going to identify them using some type of antibody testing. But we can grow viruses in a lab to help identify them. Other reasons why we can grow viruses in a lab is to make vaccines. Vaccines are modified viruses. Modified so they don't cause disease, but before we modify them, we first have to get the viruses. So we have to grow viruses to modify them to become part of vaccines. Otherwise, we grow viruses for research purpose. We want to know, how do they reproduce? How do they find their certain cells? If we know all this, we might be able to create new vaccines or stop viruses from causing different human diseases. Now, when we grow viruses in a lab, we can't use a normal plate with agar and nutrients in it. These are organisms that are intracellular, which means we have to give them a cell. And so one process that we can use to grow them is called in vitro, is we are growing them in cells. We can grow cells in a lab. We've done it. We can grow cells. We can give cells nutrition. We can grow cells, and then we can add the virus to the cells to then grow the virus. Another, in vivo, means we're going to grow these viruses in a living organism. We can use bird embryos. It's a sterile environment. They have all the needed nutrition that's going to make a virus very happy. So we use bird embryos to grow viruses. And so we can grow them in chicken eggs. It again is a reason why vaccines can be grown in chicken eggs. Why they ask you if you're allergic to eggs when you get certain vaccines. And we can also use live animals. Again, we run into ethical issues for using live animals, but depending on what we're studying, sometimes it's where we can grow the largest number of viruses, ones that we know are going to infect animals that might be very similar to us, so we can do study on them. This is just showing that we can grow viruses in cells. Normally cells are grown on plates with usually some type of nutrient media, And then as we grow the cells up, we can add viruses to them. Anywhere where you see all these blockages, that's where viruses have grown in those cells and they have destroyed the cells. So you end up with these little empty areas where the viruses are growing because they're destroying cells. Again, we can grow them in eggs. Those eggs have lots of nutrients in them. It's all one cell. An egg is one cell. And we can grow massive amounts of viruses. Now, medical importance of viruses. Well, holy cow, they cause a lot of different diseases, as we're going to come up to in our next chapters, some of the top ones that cause human diseases. They cause billions of viral infections per year. It's a guaranteed you've had a viral infection this year already. Your simple cold is a viral infection. Some viruses have high mortality rates. Those are the ones we're most concerned about. Some have chronic afflictions that, we know it causes, but we're not always sure on how to stop them. But viruses are major participants in the Earth's ecosystem. They affect not just humans, they affect other animals, they affect other plants, they affect even bacteria. So viruses can be found affecting just about anything out there. Now, detecting them and treating them gets harder. They are more difficult to detect and treat than other organisms like bacteria or some type of parasitic worms. So when we're trying to identify what a patient might have, we kind of have to look at, the big picture. Have they been around anyone that's had similar symptoms? Have they gone anywhere? Do they have any unique hobbies or, places they've traveled? And we kind of have to take an overall clinical picture of what's going on. Otherwise, to try to identify it, yes, we can try to grow these up in cell cultures, looking for specific cytopathic effects that might identify it. That can take a while. Sometimes we can screen for parts of the virus. We might be able to screen for the DNA or the RNA of the virus. Or we can even screen for the immune response. Do you have antibodies against the particular virus? They are specific antibodies for the virus. We can look for those specific antibodies. And so even right now with the COVID-19, the test they're looking for diagnosing is reverse transcription PCRs. They're looking for the DNA of the virus. And so if the DNA is present, it's because the virus is present. However, if we want to know if you've ever been infected with COVID, because maybe you were asymptomatic, we can screen the body for antibodies. It doesn't tell us if you're having an active infection, but it can tell us if you've been exposed to a particular virus. Now, antiviral drugs are also a little trickier. Can't use an antibiotic. That's specifically for bacteria. We do have a few antiviral drugs on the market. Most of them, however, can cause serious side effects. And again, it's because you're trying to target these viruses, but as you destroy viruses, you might be destroying the cells where the viruses are in. And so we do have some antiviral drugs. We're working on more of them. We're trying to stop them from sticking to cells. We're trying to stop them from being released from cells. We're trying to stop them from reproducing inside of cells. So we're trying to stop that whole replication cycle if possible. Now, the last thing I'm going to talk about are prions. And the reason why we talk about prions in the chapter we talk about viruses. Well, viruses aren't alive. They're not living cells. Neither are prions. So viruses are made up of protein, that capsid, and nucleic acid. A prion is made up of just protein. It's just protein. It's an infectious protein. And that's where we get the name prion. A prion means an infectious protein. Now, prions, it's a protein, and so as you should already know, the shape of a protein is tied to its function. And if you change the shape of a protein, you denatured it, and it can no longer do its job. Well, a normal protein called cellular protein P, PRP, is found in the nervous system. This is a normal protein found in our nervous system. However, the structure can change. We have these little helical shapes. If the helical shape stretches out, they form what are known as beta sheets. Well, that change caused a denaturation, which means it can't do its job in the nervous system, and it causes damage to the nerves and it causes damage to brain tissue. Now, when it causes damage to nerves and brain tissue, that's bad, and it actually causes holes. They're called plaques and vacuoles found in our brain tissue. Now, that's never good to have holes in your brain tissue. It's a condition caused or called spongiform encephalopathy. Pathy means disease, encephalo means brain, and it truly means a disease where your brain looks like a sponge. That's never good. It is a fatal neurodegenerative disease. It slowly breaks down your nerve tissue until death happens. Now, it is slow. It could take years, sometimes decades, before it becomes fatal, and years before you might even have any symptoms that something's wrong. Problem is, the damage is done, and we don't have any treatments for it. And it's also really hard to get rid of. Your normal autoclave is not going to destroy this prion. It's not a living thing. It's an infectious protein. It's a protein that can change shape, but it's considered infectious because once it changes shape, it encourages other proteins to change shape. That's where the infectious part causes. It's kind of the bad influence. It changes shape and it encourages everyone around like, hey, it's so cool to have these straight beta sheets. And it encourages other proteins to change shape. Now, this is just showing how you can have these infectious proteins that, yes, when they start to change shape, they encourage other normal proteins, and they all change shape, and they start to cause damage to the neurons, and they cause damage to the nervous tissue. Now, different diseases that this can cause. Most of them are animal diseases. Something's called scrapie in goats, transmissible mink encephalopathy. Most common ones you've probably heard of is chronic wasting disease, because we have that around here in deer, and the mad cow disease known as bovine spongiform encephalopathy. And again, it's a disease that causes the brain to look like a sponge in cows. Now, it's called mad cow disease is because obviously if your brain isn't working correctly, it's under attack, they're going to act different, and that's where the mad cow disease came from. However, there are some human prion diseases. Creutzfeldt-Jakob disease. I don't even want to try to pronounce these. The kuru has kind of gone away. It was found in some African tribes. But in the African tribes, one of the ways to honor their dead was to eat the brain tissue of the dead. And that's where it was passed on. They discovered that. They stopped doing that. Kuru is now gone. But there are some human prion diseases. The big question is, can an animal prion disease cause human issues? Well, there are cases of humans getting mad cow disease from eating infected beef. Because again, even muscle tissue has nervous tissue in it, running through it. And so there are cases that, yes, humans have picked up this prion from eating beef. So the big question around here is, can you pick up chronic wasting disease from eating infected venison? I don't know. It's an awesome question. Problem is, it's pretty new. Chronic wasting disease is pretty new around here, and because this is a slow progressive condition, we might not know the answer to that for several decades. And by the time we know an answer, it's too late. Damage is already done. Now, my last concept check question, exposure to nucleases, so enzymes that break down DNA and RNA, would damage all of the following except, well, it's prions, because prions are just proteins. They have no DNA or RNA. It's just an infectious protein. So we're going to end right there. Again, if you have any questions on virus basics or on prions, please send me an email.